Today I'm going to talk you through the process of glycolysis and all of the steps involved. So to start off, we have a glucose molecule here represented by these 6 orange circles, meaning each circle actually stands for a carbon molecule. This 6-carbon sugar is first of all phosphorylated. So this is when we attach phosphate groups to the 6-carbon sugar, forming hexose biphosphate. Hexose meaning 6-carbon sugar, and bi 2 phosphate groups. As you can see in this diagram, the phosphate groups are actually represented by these pink circles here. Where do we get the phosphate groups from? So these 2 phosphate groups actually come from 2 ATP molecules, which give up 2 phosphate groups, 1 each, and that leaves us with 2 ADP molecules. The next step is lysis. So this is when our hexose biphosphate is split in 2, because that's what lysis means. It means splitting of a molecule. So our hexose biphosphate has been split and it's formed 2 triose phosphate molecules. The 2 triose phosphate molecules are then oxidized in the process of oxidation, and at this point we also have 2 NAD plus hydrogen carrier molecules come in, which are reduced to form 2 NADH plus hydrogen ions. This leaves us with 2 triose biphosphate molecules. As you can see, triose 3-carbon sugar with biphosphate 2 phosphate molecules attached. We then undergo ATP formation, which is the final step, and this is where we bring in 4 ADP molecules and we remove these phosphate groups and attach them to the ADPs, forming 4 ATP molecules. Finally, we are left with our 3-carbon compound pyruvate, but don't forget we've still got 2 of them represented here. In summary, what we have here is we have 1 6-carbon glucose molecule coming in and 2 3-carbon pyruvate molecules coming out. We have a total net gain of 2 ATP molecules. As you can see, we have 4 ATP molecules coming out here, but we also put 2 ATP molecules in at the beginning, and then we also produce 2 reduced NAD molecules, which are then going to be used later on in other stages of cellular respiration.